beta sheet or alpha helix. Yeah. Tertiary structure is how, again, how the three-dimensional structure, the folding of the proteins are interacting, and quaternary are subunits. Okay? It's very important that you know that the structure of a protein that is with the sequence of amino acid, well, we discussed, right, that the type of bond that interact between one amino acid and another is the peptide bond. And this is a very strong interaction, right? It's covalent bond. We're going to see that for the secondary, tertiary, and quaternary structure is not the case. We're going to have a lot of, for example, hydrogen bonds, etc., that are easy, right, to break apart. And here is, so primary structure, you're going to have the sequence of amino acid, right? When you're looking for any protein, the only thing you're going to see is this. And what is this? You can tell me what is this? MIKKIGB. What is this?
It's the single letter abbreviation for each amino acid.
Correct. What we have here? What is M?
Methionine.
Then?
Isoleucine.
Then?
Lysine.
Lysine.
Then? Isoleucine.
Okay. And so on and so forth, right? You don't have to say everything. But yeah, that's when you're looking for any protein, and I put this example, it's not for you, I mean, just for you to know, right? If you go to the NCBI, which is a website from the NIH, right? It's a database of any protein that you're looking. I put here DAF5 because it's the protein that I work with, sorry. I put DAF5 and look at the amino acid. It's a pretty big amino protein. How many amino acids does DAF5 have? How many amino acids do you see here?
781.
700.
Are you asking how many amino acids?
901 here.
Oh, sorry, I was just looking at the bottom line.
Yeah, this is 901 and all of these. So someone asked me in the previous class, like, oh, what is that number? 45, 43. That's the sequence of amino acids. So if I say, well, let's pretend that the catalytic unit of this protein is in the amino acid number, I don't know, 100. So I just look for 100 and that's it. There's a mutation, well, I can look at it and, you know, and work around that. So that's what basically when you are studying proteins, it's very common that you should know what is the sequence of amino acid. And it's always from the N terminus to C terminus, okay? Again, we covered that, the peptide bond, right, which is a condensation reaction or dehydratation because we have water as a product. And that's the type of bond that bind one amino acid and the other amino acids. You should recognize how it looks, the peptide bond here. Here's 1, 2, 3. This is, if you have 3 peptide bond, that means you will have 1, 2, 3, 4 amino acids, right? Okay. So the characteristics of the peptide bond is considered as a double bond. And if you recall from your organic chemistry, when you have a double bond, it's very rigid and cannot rotate. So this peptide bond, this is basically the representation. It's like a double bond. So when you have the peptide bond between the amino acid, you cannot rotate. However, the R group, which is that represent each amino acid, and I think I forgot to mention that, but R, they also call residues. So residues is each amino acid, right? So the R is what is different between each amino acid, okay? This part is rigid because this is the double bond, but this part here, the R groups are freely and they can rotate, and they actually rotate and they can do the secondary structure, tertiary, and quaternary, okay? And what else from here? Okay, so the angle, there's some angles between the peptide bond and the end group and the peptide bond and the carboxylic group. They call it phi angle right here and C angle right here. What do you have to know from here? Okay. There was this scientist called Ramachandran, and he actually studied the angles of the amino acids, right? Of the proteins. So he realized that each angle behave differently, and you don't need to know how to read the Chamandran plot, but you should know that this is useful to project how a protein will be if they have more beta sheets, alpha helix, etc. For example, if you have glycine, you can have, this is a Ramachandran plot, you can have beta sheets, you can have right-handed alpha helix, etc. If you have proline, and if you recall proline, it's the only amino acid that the R group is bind to the end terminal of the amino acid, and it's very rigid, and you cannot find a lot of rotations between proline. Okay. So basically, for each protein, you can have a projection for the Ramachandran plot. For example, this enzyme, which is involved in one of the last steps of glycolysis that we're going to study at the end of the class, of the course, is pyruvate kinase. So you can see here that there's a lot of alpha helix, right? And some beta sheets. And basically that you can't, based on obviously in the sequence of amino acid, it can project how it will be, the 3D structure of that protein. So that basically is the Ramachandran plot. Okay. So the secondary structure, we can have alpha helix, that is this. We can have beta strand, which is this. And we can have beta turns and loops. Okay, so let's see one by one. Oh, so every time you see this type of a structure, right away, you should know that are proteins. And we're talking here about the secondary structure of the proteins. Okay. So, okay. I'm going to put a video, but first, this is the alpha helix. As I mentioned, the primary structure of the proteins are the sequence of amino acids. The secondary is, as you can see here, for example, is the helix. The residues, right, of the amino acid that are not too far apart, but far apart, they can interact because there is a change, right? It can be alpha helix. See? So there is a twist. So therefore, this amino acid right here, each R is the residue, right? It's each amino acid. So this amino acid that is here, you can see here, is interacting with the other that is far apart. Okay? So here is just saying that for each turn, there are around 4 amino acids. Okay? And the type of bonding that you have in secondary structure are actually hydrogen bonds, as you can see here. Okay. Here is just showing, depends, but sometimes in alpha helix, you can have hydrophilic amino acid outside and hydrophobic amino acid in the interior. Okay. Before I continue, I will put a video so you can visualize and look at how the secondary structure will be, but the majority are alpha helix and beta sheet, and the type of bond or interaction are hydrogen bonds, okay? So, questions for now? I'm gonna put the video and then if you have questions, we can discuss that, okay? I think I'm not sharing.
The three-dimensional structure of a protein is defined by 4 hierarchical levels. The primary structure refers to the amino acid sequence of the protein. A protein's secondary structure describes the arrangement of local regions of the polypeptide backbone. Tertiary structure refers to the spatial location of all the atoms in a polypeptide chain. Finally, quaternary structure describes how different polypeptides are arranged within a complex. Almost all proteins contain combinations of 3 secondary structure elements: alpha helices, beta strands or sheets, beta turns, and loops. Alpha helices are right-handed helices that place the amino acid residue side chains on the outside of the helix relative to the polypeptide backbone. The alpha helix structure is stabilized by intrastrand hydrogen bonding between the carbonyl oxygen in one amino acid and the hydrogen of an amide nitrogen on an amino acid 4.
You cannot hear the video?
We can.
Ah, okay.
It was frozen, but it got fixed.
Oh, okay, okay. Let me put it back.
Relative to the polypeptide backbone. The alpha helix structure is stabilized by intrastrand hydrogen bonding between the carbonyl oxygen in one amino acid and the hydrogen of an amide nitrogen on an amino acid 4 residues away. Many alpha helices are amphipathic, meaning that one side of the helix is hydrophobic and the other side is hydrophilic. This property is important since alpha helices often need to interact with both the hydrophobic core of a protein and the aqueous environment at the protein surface. Amphipathic alpha helices are also able to pack closely together with their hydrophobic surfaces facing inward or outward, depending on the chemical environment of the protein. Another common type of secondary structure is called the beta strand. A beta strand is made up of an extended polypeptide chain where adjacent amino acid side chains alternate, being either above or below the polypeptide backbone. Multiple beta strands can form beta sheets through interstrand hydrogen bonds. Beta sheets can form through interactions of parallel beta strands or antiparallel beta strands. Beta sheets are rarely flat and are often twisted to various extents within the context of a protein. The third category of secondary structure found in proteins include beta turns and loops. A beta turn is a short 4 amino acid segment that connects 2 beta strands in an antiparallel beta sheet. Loops are irregular segments that often connect elements of secondary structure and are usually found at the surface of proteins. Through understanding the three-dimensional structure of proteins, we can understand their function in the cell.
Okay. So now you can have an idea, right, how it looks like. And again, the secondary structure is basically the interaction of the R groups that are far apart, but not that far apart, right? Important hydrogen bonds are really, really important to connect one amino acid and the other right here. Again, this is represent each amino acid. This is just showing that it's four amino acids that are in each turn itself. Okay. So for example, these are group is one amino acid that is interacting with the one that is over here. Okay. So there's a twist, and that's how the alpha helix, right? So certain protein will have a lot of alpha helix. So for example, well, we're gonna cover some of the examples. Okay, and this is what the video show you, right? That some of them, some alpha helix will have in one of the side will be hydrophilic and the other hydrophobic. Okay. Here's just showing that some amino acid will be predominant in the alpha helix. I'm not gonna ask you exactly what are the amino acids that are present in alpha helix, etc. But it's just for your knowledge, right? Some of them are more predominant than the others. Glycine is very tiny, right? It's very easy and it can be accommodated anywhere. Proline is a little bit more complicated. What is the question? Which amino acid I'm going to show you right here. I'm going to go back. But for example, there is a table. Again, I'm not going to ask you what is, but some amino acid predominant in one of the conformation. For example, alpha helix, it's telling you, for example, that methionine, glutamine, alanine, these are predominant. Beta conformation, well, you have more valine, for example, beta turn, etc. Right? Some of them are more, the probability to find this type of amino acid are depends, right? Again, I'm not going to ask you in the exam what are the amino acids that are in the beta conformation, but the information is there if one day you need it. Okay. The important part here, for example, is to know that this is a secondary structure, right? The alpha helix and beta strand are secondary structures. So every time you can see here this structure, you know that is a protein. And their protein, as I was mentioned, there's some proteins that are just alpha helix. Very simple. And that is the case of a very important protein that is present in our hair, which is keratin. So this is the secondary structure of keratin. There's a lot of alpha helix in there. It's considered a fibrous structural protein, and it's just alpha helix, okay, right here. This is an example when the hair is curly, there is a lot. That's the reason. The reason that why it's curly is because it contains a lot of cysteine. This you should know. The cysteine, and we cover that. The cysteine is one of the two amino acids that contain a sulfur, but these actually can react one cysteine with the other. There's an oxidation. Okay, so when you have a curly hair, you have more presence of cysteine. So when you straighten the hair by treatments or heat, what you're doing basically is breaking apart these disulfur interactions between cysteine. Okay, this is basically the reason. Okay, this is the other type of secondary structure, which is the beta conformation. If you see something like this, again, that is the secondary structure of a protein. The video mentioned that, and you can see the video in one of your homeworks. Some of you already complete the homework, which is great. You can review the video if you want through the homework. Again, hydrogen bonds are very important. And the secondary structure, well, there is interaction between residues that are apart. So these interact with these, with hydrogen bond, etc. It can be parallel right here or antiparallel. This is just how the interaction, how they interact, then it looks like a sheet, right? Like from the bed, right? A sheet, a piece of sheet. That is a type of secondary structure. Okay. A protein that contain a lot of beta strand or beta sheets are silk. This, for example, here, this is a protein that it get, right, strain, right? It's very, very strong protein itself, and the conformation is beta sheets. Beta turns, basically, something that you should know, proline and glycine are predominant here because, well, glycine, again, it's very small. Therefore, it can be accommodated in many of the structure, but beta turns, you can actually find it. And proline, again, proline is the only amino acid that the R group is bind to the N terminal, okay? Yeah, you should know the 20 amino acid. They're asking if they should know the 20 amino acid. Yes, the characteristics of each one, okay? And I show you these, some are more predominant than the others. Okay. Something else that you should know, what is the difference between motif and domain? Motif is the structural, in proteins, a structural motif describe the connectivity between secondary structure elements. Those are not stable itself. For example, helix, loop, helix. Helix we know is a secondary structure, right? But when they're connected like this, we call motif, okay? And the proteins, they have motifs. It depends on how the conformation it is. For example, we say, oh yeah, the protein that I'm looking, it's a beta alpha beta motif. See, it depends if it had more helix or beta strain, etc. Okay. The domain means it's a specific segment of the protein that have a specific function. For example, transcription factors contain a DNA domain. So the part of the protein that is in charge of interacting with the DNA, that's the domain itself. And it say that it can function itself because that's the part that is in charge of creating, for example, the interaction between DNA and the protein. Another domain that is very important is the ATP domain, right? So when you hear that, it's because that is a segment of the protein that is in charge of doing that specific function, okay? So DNA domain, ATP domain, etc. It's very common. Let's see what else. Questions so far? Good. Okay. Okay, about tertiary structure, that's it. So we covered primary, secondary, now is a tertiary structure, okay? The tertiary structure is when they interact, when they're far, far away, right? Is the folding of the protein itself, okay? And this is an example, right? So the 3D structure is when the protein is already full and ready and functional, totally functional. There's several interactions that occur in a 3D structure. One is hydrogen bonds. We have disulfur bridge, between cysteines, we have ionic bonds, and we have hydrophobic interactions. So there is not a single one, there's several. Like in the second one, in the secondary structure, the predominant were hydrogen bonds. Here you have more, and that's how the protein is going to be full and can be functional, right? In the case of sickle cell disease, the folding was not correct because of that amino acid. It's because the folding, that amino acid specifically is negative. It is necessary for the correct 3D structure of that protein itself, okay? So there's several type of interactions, not just hydrogen bonds, okay? The Protein Data Bank is another website, right, that you can go and look for any structure of any protein that you're looking for, all the information, the crystal structure, the X-ray, all of the information from all the proteins are there. Okay, that's just for you to know. Okay, the proteins can be fibrous or globular. The fibers are very abundant. We're talking about keratin, silk, collagen. And the globular are globular itself, and one of the best examples are immunoglobulins, right? The antibodies itself. Okay. An important thing about proline, again, we have proline again and glycine again. Proline actually can be modified, and this is very important for collagen. Collagen is a very abundant protein. It's basically imagine it's the glue, right, from many of the things in our body, the bonds, etc. Actually, proline, it is modified, right, by this proline hydroxylase. So that means it will add a hydroxyl group, right, this OH group. And this is important for the conformation of the collagen itself. So back in the day, a long time ago, they find out that actually the pirates, right? The pirates suffer from diseases, several diseases, you know, the teeth and the skin was really ugly, blah, blah, blah. And they figured out that the problem was when they went away in this, you know, in the sea, etc. And they have this, they went away for months and months without fruits, without vitamin C, etc. They formed this disease, which is a scurvy. That doesn't exist anymore unless you have a deficiency of vitamin C. And vitamin C is actually a cofactor for this protein to add the OH on proline. And this type of modification in proline is necessary for the formation of collagen. So after that, they figured out that the only reason is because the lack of vitamin C, which is a cofactor for that specific protein. Okay. Disulfur bonds, I mentioned, is very important for the 3D. Again, people who have their hair a little bit curly is because contain a lot of cysteines. The way to remove, right, to have the hair straight is by applying a heat or chemical that disrupt the interaction between the disulfur cysteines, right, between one and another. What else? Well, here, if you look, you should identify right away that these are helix, alpha helix. Those are turns. Here's a mix, right? We have alpha helix, but also beta sheets, etc. So for now, every time you saw this type of a structure, you should know right away we're talking or we are referring to a protein, a secondary structure. Okay. Okay, quaternary structure is basically a subunits that form a protein. And not all the proteins will have a quaternary structure. Again, primary structure is the sequence of amino acids. Secondary structure is the alpha helix or beta sheet. Remember, it's an interaction between the primary was right away one and the other. The secondary, it's interaction between amino acids that are far apart but not that far apart. And the tertiary structure is when they interact, the 3D folding conformation. Quaternary are subunits. As you can see here, are subunits that interact together and they can form a protein. An example is hemoglobin. Hemoglobin is this huge protein, right? It's a homodimer because it has this, the same units, but there are subunits, right? There are 4 different units that are bind together. That's the quaternary structure, okay? We're gonna study, it's very interesting, hemoglobin, that how it binds, right, to the M group and deliver the oxygen in our body. Provide properties, well, yes, some of them, not all the proteins will have quaternary structure, but some have the quaternary structure. Yes, Nicole?
So did you say that hemoglobin is a homodimer, and then its quaternary structure is a few of those homodimers together, and that's why it's 8 parts?
No, it's 4. It's
4
home
parts.
because it's the same structure. When it's heterodimer is when they're different.
Oh, got it. Okay, thank you.
Any other questions? Okay. Something very important is protein folding. I mentioned that one of the reasons the sickle cell disease is because hemoglobin cannot fold in the proper way. So folding, right, in here, you should know this term, right? When the protein is unfold, it's, not fold. It's not functional. It needs to be fold in order to have all the 3D structure in order to function and do whatever the protein it's in charge of doing, right? When you have a folded protein and you disrupt all the interactions of hydrogen bonds, ionic interaction, disulfur bonds, that is denaturation. When you denature the protein, this is a term that we use, right? When you disrupt the folding of the protein is not functional anymore, right? The classic example when you cook an egg, right? The protein from the egg, well, with the heat, you disrupt the protein itself, okay? Here's just showing that it suggests that when it's folded, the delta G will be negative, meaning is favorable, right? Here should be delta G, not G. So then it should be favorable for the protein, and then when it's folded, then that means it's active, okay? The denaturation I mentioned is you lose the 3D dimension of the protein. When you denature the protein by heat, by different chemicals that disrupt all the interactions that you will have in the 3D, present in the 3D, we call denaturation, okay? If a protein is denatured, that means it's not functional anymore, okay? It's inactive, basically. From here, what you should know is, well, Amundsen won the Nobel Prize. Well, there's a lot of Nobel Prizes here in chemistry. By the way, the day you present your exam, I think they're gonna release who is the winner for the Nobel Prize in chemistry. So we'll see. Anyway, here, the only thing, in this experiment, basically, what happened is that these RNAs, which is a ribonuclease, and if you recall, and this you should know, nucleases are the one that chop they're like the scissors, right? And chop and cut the nucleotides, in this case, ribonucleotides. What he did is basically, well, he said, well, what happened if I add chemical that disrupt, right, the like methylketotenol that disrupt the disulfide bond? And what happened is that obviously obviously now, right, the protein is inactive. However, when in this case, when they were able to put together again those disulfide bonds, what happened is that the enzyme was able to be active again. And basically the whole thing here, and he won the Nobel Prize, is because he discovered that actually if you don't disrupt the amino acid, the primary structure, your protein can be active again. Okay? Unless you disrupt the primary structure, which is the sequence of amino acid, your protein can be active again. Okay? So here is inactive because, as you can see here, it's different. You don't have the disulfide bond that you need for the 3D and the folding, the proper folding of this protein. But when you have the correct disulfide bond, then the enzyme is active. Okay. This is basically the same. This is catalytically active. You disrupt that interaction, which is essential for that protein. Then it's unfolded and then is inactive. If it's possible to put back those type of interaction in the 3D, then you have a protein that is active. Okay? Okay. There's several models for folding. One of them, and I'm not going to ask you, there's three. I'm not going to ask you, what is, the framework, etc. Just to have you have an idea. It's important, it's essential, the protein folding, absolutely, because that's the way that they're going to be active. In this hydrophobic collapse model, it's suggesting that the amino acids that are hydrophobic, they go and fold first, and then the rest are going to fold after that. The other suggests that is randomly the way that the proteins are folded, etc. But the important part is that the protein need to be the correct folding of the proteins. The delta G is negative because it's favorable, and unless you disrupt the primary sequence of the amino acid, your protein should be fine. Okay. And what is the denaturation? There are certain proteins that help the folding of other proteins that I cannot do it by itself. Normally, the proteins can do this by mechanism by itself, but there are others that they cannot do that. And chaperones are proteins, and yeah, you should know that. The chaperones are proteins that will help. We're going to see a video. It helps and uses ATP to help the folding of the proteins. Not having a correct folding, again, cause diseases like sickle cell disease. Alzheimer is another type of disease just because the protein cannot be fold correctly. It's protein misfolding. Okay, so they can actually cause diseases. So it's very important that the protein folding is correct. The amino acid are the amino acid that they should be there. If not, you can have consequences of like diseases. Okay, questions? I'm going to put the video for the chaperones, and that will be the last thing or the stopping point for your exam. Okay? Questions, comments? We good?
Professor,
Yeah.
What you teach today is going to be an exam?
Sorry? When is exam?
No, this that you're teaching today now is going to be an exam?
Yes, today is the stopping point. Yes.
Okay.
In order to be functionally active, proteins must fold to give rise to a specific three-dimensional structure. Although many cellular proteins are thought to fold successfully on their own in the cytosol, some proteins appear to require the assistance of special proteins known as chaperones to complete the folding process. One of the best characterized chaperone proteins is heat shock protein 70, or HSP70. HSP70 is a clamp-like protein that, when bound to ATP, undergoes a conformational change that reveals hydrophobic groups, allowing unfolded or misfolded proteins with exposed hydrophobic residues to bind. When ATP is hydrolyzed, HSP70 closes on the misfolded protein. After ADP is released, HSP70 opens to free the partially refolded protein. This cycle can be continued until the protein is completely folded. The GRO-EL-GRO-ES complex is an example of a bacterial chamber-like chaperone. The GRO-EL subunit consists of 14 identical subunits arranged in 2 rings of 7 subunits each. Each GRO-EL subunit has 3 protein domains: a substrate binding domain, an intermediate domain, and an ATP binding domain. The substrate binding and intermediate domains form the walls of the chambers, while the ATP binding domain separates the upper and lower chambers. GRO-ES also forms a 7 subunit complex and functions as a cap for the complex. The GRO-EL-GRO-ES protein folding cycle consists of 4 steps. First, GRO-ES and 7 ATPs bind to the upper GRO-EL ring and trap an unfolded protein inside the chamber. Then, in the lower chamber, a conformational change occurs that releases GRO-ES, 7 ADPs, and a partially refolded protein. After a few seconds, the 7 ATPs in the upper chamber are hydrolyzed, causing a conformational change in the upper chamber that facilitates protein folding. Meanwhile, the lower chamber accepts a new unfolded protein. In the last step, 7 ADPs, GRO-ES, and the partially folded protein are released from the upper chamber, and the cycle repeats itself. An estimated 30% of protein synthesized in the cell must be refolded or degraded. If left to accumulate, misfolded proteins can aggregate, interfering with cellular processes and potentially leading to disease.
Any questions or comments? Yeah. So this will be the last, the topic that will be in the exam. Oh, it's early. Okay. You have questions, comments?
Professor?
Yeah.
Are you going to be doing review on Monday?
On Monday, I will be addressing any specific questions or topic. Yes.
Okay.
Any other question?
Professor, this last video, is it saying that if there are too many proteins in the body, it can cause disease?
No, so the video was about chaperones, which are proteins that help the folding of the proteins. If the folding is not correct, then they can cause a disease.
So it's just the folding, it's not too much protein. Okay.
It's the protein, is the folding, yes. And I think it's in the, if you want to review it, it's in the homework that it's due it on Saturday.
Okay, thank you.
If you don't have more questions, that's it until the exam. It's 4 chapters. On Monday, yes, I'm going to address any topic you want me to repeat, or questions, specific questions? Okay.
Could you go over motif versus domain one more time for me?
Yes.
Thanks.
It's not this one. The motifs are how they are arranged, the secondary structure. Remember, the secondary can be alpha helix or beta strand, etc. So sometimes they are together. Well, they are together, the whole thing. So that's how is the connectivity. Some of them are those motifs are common. For example, you would say, well, my protein. This is my protein. My protein is a helix loop helix. So this is a very a normal vocabulary, right? When we're talking about proteins, because that means that my protein, the protein that I'm studying, has a lot of helix and then a turn and a helix. So it's called helix loop helix. Okay. If they are by itself, they cannot be. They are not independent as the domain. So the domain basically is the segment of the protein that has a specific function. I mentioned, for example, the DNA domain. Again, for example, if I'm working with a protein that is a transcription factor will bind to the DNA. So that segment is the DNA domain, and that's how it's called. So for example, again, if there is a mutation or whatever in the DNA domain, there is an issue, right? Because if the protein is in charge of binding to the DNA and there is a mutation in the DNA domain, it cannot bind. So the domain itself can do a specific function. In this case, bind to the DNA. We have the ATP domain. Enzymes that are kinases, they have the ATP domain, meaning they have a segment that are specific involved in the ATP. For example, kinases transfer a phosphate to another protein. Okay, that's the difference. So the motif is just how the secondary elements, secondary structure are together. And the domain is the region of that specific protein that has a function. Okay. That's why it is said that can fall independently in a stable 3D structure. Okay.
Okay, thank you.
Any other question?
Professor, this is Joanna. I sent you an email. Did you receive it?
Yeah, I replied to you.
Oh, okay. Okay. I haven't checked my email because I was at work all day, but I'll check it. Thank you.
All right. Any other questions? If you don't have questions, go and study. Okay.
Good night, Professor.
The same, and see you. If you have questions, email me and I will see you on Monday, okay?
Thank you, Professor.
Bye.